Thank you for inviting me, and thank you for hosting so many wonderful people in a place I called Science Candyland back home. The Allen Institute is awesome, and you're doing a lot both here supporting educators and through the website with valuable resources. So I hope we can continue playing. I like playing with puzzles, while my wife likes physical puzzles. I don't like those because we lose pieces and the dog eats pieces and then you get frustrated. I like puzzles that are more on the bioinformatics side, microbial genomes. And this was something that I fell in love with, learning that we can do so much on a computer with sequences that are now publicly available. And these puzzles seem simple, but they really get more and more challenging as we add more players. And we have the opportunity now to look at microbial communities and not just sequence one genome, not just one puzzle, but multiple puzzles at the same time. And deconvoluting and making sense of this is a challenge that I've really enjoyed. And this has been in partnership with a fantastic team at the KBase Educators Group. I'm also sharing the slides at a short URL, go.ncsu.edu, and we can talk later if wanted. But one thing that I've been intrigued by was metagenome assembled genomes, this notion of trying to put together the individual puzzles in a mess of multiple puzzles. So, for example, you may have hundreds or thousands of different bacterial genomes in an environment, and now we can sequence the DNA, and we want to put it back together. And how do we do that? And this graphic over here is not just because I'm obsessed with the Noun project, but it also symbolizes this, and I use it with students in classes. So we may be able to put together 3/4 of a genome for one organism, a full genome for another one, something linear for another one. So they are really not full genomes, yet they have valuable information. And conveying this to a class of undergraduates and graduate students, first and second year students now, is something I've been intrigued by. So today I'll tell you about why, who, what, and how. So why teach the details about bioinformatics tools, who and what is the KBase, the tool we're using. We'll talk a little bit about what are MAGs and how do we teach about MAGs using a variety of tools. So we use Annotation, we've used Hypothesis and Perusal, KBase narratives, and publicly available databases, and we do some assessment with surveys, data, and analysis. And I'll leave you with food for thought because I don't have many results. This is a project that I've tried to convince others about, and they've run away, several grad students and postdocs. Yet I'm convinced there's something interesting here. So why teach the details about bioinformatics tools? We're biologists. I often have geneticists, crop science, chemical engineers in the same class, and I've thought about this. Should I be teaching the details of the tools we use, the bioinformatics tools, or should I leave it as a black box? I am not a computer science scientist. I dabble in bioinformatics, but I don't know some of the details, and that literature is dense for me to understand. So a while back, and this is a fairly recent article that was published, has a quote that made me think. As life sciences research has become more dependent on complex and abstract computational tools, such as machine learning and AI, the need for those in the field to have a basic understanding of the appropriate use and interpretation cannot be overemphasized. And I read this and I stopped and I said, well, I guess what we're doing is not just subjecting students to the details of the methods, but hopefully understand the process. So our research question, and I'll describe the hour in a couple of slides, but our research question stemmed from a conversation I had with a research postdoc back then at NC State. How does learning the computational methods affect the conceptual understanding of metagenomics? So conceptual computational methods, or the computational details, and then we have really the biology concepts. So what we did was use this tool called KBase. And KBase is really neat. It saved me during the pandemic. A group of us met monthly and developed a set of tutorials for educators. The KBase team is fantastic. Ellen Dow and Alicia at UC Berkeley. But basically, it's a web-based platform by the Department of Energy. You can create interactive notebooks that are Jupyter notebooks that are shareable, reproducible. You can save static forms, and these have access to powerful computational tools without the command line and data sets that are publicly available. So it makes a really great tool for community members to share resources and for teaching some of the principles of fair and open data sets. As part of the KBase team, they've developed a microbiome workforce development program. And these 5 steps on the screen are really impressive work that Ellen Dow has done. So they started with a research question and encouraged educators to pursue their own research questions. We developed ways of collecting metadata with the National Microbiome Data Collaborative. We partnered with companies and Neon to standardize protocols, get some discounts on sequencing. And KBase developed some really great tutorials for teaching with KBase. And the last one is one I really like. They've partnered with the American Society for Microbiology and Microbiology Genome Research Announcements so that educators and classes can develop and analyze and sequence microbes and have publications with students. So this is an example on the website of the journal saying they have a collection of student and educator produced publications, genome announcements, which I love. And this is a publication. Anna McLuhan, who's fantastic, is the first author at a smaller institution in the Northeast. And everyone else, except the last one, they are all students. And they are announcing 5 draft genomes that they got from the parking lot of their science building and isolated and then enriched and learned more about. What's nice about the platform is that you can save the data. So this is, for example, one of Anna's narratives that's in static form. So this is locked in time, has a timestamp on the top right, and you can see each step of the data processing. So it's great for reproducibility, and it's great for a teaching tool because we can revisit those narratives and say, look, this class did this, what are you going to do? But we are complicated. Anna is looking at single isolates, one bacterial organism grown, enriched in, for example, wetlands, or in the class that I teach, we look at electronic waste. We take feeding tubes from waxworms. We get those microbes. We enrich in media that has discarded electronics from North Carolina State. And then we look for the microbes that can degrade the plastics in printed circuit boards. And these are not just one isolate, they are many genomes. So what are MAGs and metagenome assembled genomes? Here comes Jason Whitham. He was a postdoc that joined, finished another postdoc at Oak Ridge National Labs during the pandemic and wanted to gain teaching experience. And turns out Jason is a real expert in the wet lab stuff as well as the bioinformatics, and was stolen by Takara to do research with the company after doing a postdoc at NC State. But during his postdoc, he introduced me and the class to the concept of assembling microbial genomes or reconstructing them from lots of genomes in one environment. So trying to make sense of that convoluted data and assemble the pieces, the individual puzzles. So how do we teach about MAGs? We do it in a very structured way, and this was something Jason and I talked about. We first read and discuss articles that provide the biological context, the key computational tools, and an application that's similar to the one that we discuss. And in lab, we use KBase and data from the original study, interact with the tools in class and discuss them, and apply the process to a challenging problem using an open public data set. So we use social annotation. I've used hypothesis. Our campus moved to perusal, and this is an example of the first article we read. So the first article was Jason Whitham is the second author there. He did this work at Oak Ridge. It really describes the biological context. It's published in a research journal that does microbiology. And they look at anaerobic, thermophilic, methanogenic microbes fermenting switchgrass over the course of time. And it has some complicated numbers, but it's mostly bio and genetics research. And students are encouraged to annotate. They have to make 10 comments, questions, and YouTube videos, links to YouTube videos explaining this count as one of their 10 comments. The second paper makes people wonder. So the second paper is a true computational paper we read. So this is one of the first algorithms to try to make sense of metagenomics, multiple genomes in one context, that pull out individual ones by separating things that look like they are part of one genome and not another. So this tool is called MetaBat2. This journal is a computer journal, and it's dense. It's way out there for some of our chemical engineers and biologists reading this. So again, they have had practice. We want them to annotate the article, explain to each other what's going on. And the third one is an extension. Now that you learned about this one study and this one approach, this is an application in a third setting using a similar technology. And in lab, we do this same approach in parallel, but using two narratives, two KBase workflows. The first one is made by Jason. Turns out the plant he works with is nicknamed Silver, switchgrass, Silver. So we called it the Silver narrative, and we prompt students to work 10 minutes to explore it with their group, explore the narrative. Most of the steps have been laid out in a graphical way. We explore various tools, computational tools, and we have some output. Some of the tools have been pre-computed, some have not, and they have questions, critical thinking questions as they go. So for example, this is the narrative that Jason wrote. It has instructions. He knows all the background. He adds steps, and runs us through each step. And on the left, you see an outline of, we start with analyzing the sequence data, and doing some quality control, doing some classification, and then trying to assemble the data together. And try to separate it out based on individual microbes. The second narrative is the one that I did. I'm obsessed with a gold-pooping microbe that takes liquid gold and precipitates it out into gold nanoparticles. We all love Delphtia the alchemist, and this gold narrative uses the same approach, but instead of having a data set, it prompts students to look in the databases available for a data set they think will have Delphtia. And their way of finding Delphtia is to use the approach they just did with the silver narrative, assemble the data together, and try to find Delphtia microbes. And then we prompt students to reflect, share in individual reflections and in classroom, analyze the tools, and think about contamination, how good was the approach, what are some of the limitations. So on the education side, we've collected for the last 3 or 4 years, IRB approved annotations of articles from undergrads and grad students. And mind you, my classes are 16, capped at 16s, and I teach it once every fall, this class. Jason did pre and post surveys and pre and post quizzes. So the quizzes are 16 to 19 questions. The post quiz has some extra open-ended questions. They are tough. They are questions on the computational methods, concepts, and some math involved. And the pre and post surveys are confidence levels and using a bioinformatics confidence instrument. And we've done this for a couple of years and finally got to 50 student responses last semester. And my IRB told me I could not analyze till after the academic year was done. But some things that I've accumulated over the years is an interesting trend. There is an increase in grades. So these are the quiz grades and the multiple choice conceptual understanding between the pre and post of student responses. Pre is at the beginning of the course, post is at the end of the course. So they are learning some of the concepts. There are still some misconceptions. And you see that there's some points and some students still make some mistakes. Some students actually even decrease in grades, which to me is really interesting because there are some misconceptions or additional details that are not captured by our multiple choice questions. And then we've accumulated lots of qualitative data asking students, how do you decide to analyze reading assignments this way? Why? What was your approach? How did you annotate the reading assignments? Why did you use this approach for the KBase? Why did you use this approach for annotating articles? And what I'm hoping to get is an idea of what are the challenges that prevent students from fully using the tools, the bioinformatics tools, and fully exploring some of the applications of these really powerful web-based tools. Why do we struggle with some concepts? Is that specific to the way we are teaching it? Or is that something with the way the tool itself is presented? So some food for thought for you all is, when and can we or should we create a deep dive into the methods? Do the methods help students learn the concepts? In this case, the biology, microbial genomics, and genomics concepts. What data would convince you as an educator of this approach? I've been asking students why do they use that approach? Why do they read this way? How do they explore KBase? When are method papers appropriate? When are they outdated? MetaBat 2 is now MetaBat 4. Should I be annotating MetaBat 4 with students, or should I start with the first one? When is it too complicated? I've had students say, I didn't sign up for this. This is not a computer science course. And student approaches. When do we gain more confidence working in groups? When do we gain more confidence working individually with an open-ended public data set? And I want to do some focus groups to learn from students what they are up to. So this really does help promote reproducible science in the classroom. Some of the questions that we've asked students is what do they think about open science using public databases? We ask about reproducibility of the approaches with the approaches they've learned. We have some ethics questions integrated and some authorship and data management. And with that, I ran out of time, but this was a team effort. And while I don't have any solid results, I'm always intrigued by some of the responses students provide to the open-ended questions we ask, as well as some of the misconceptions in some of the concept quizzes that we provide. Thank you.